Today we will study about the general accounts of the dicot stem. The stem consists of three distinguishing tissue systems: the dermal, the ground tissue, and the vascular system. Dermal tissue. The stem remains externally bounded by a single layer of loosely arranged rectangular cell called epidermis. It develops from the dermatogen. Epidermis dermal cell is inaquatic and shade-loving plants are thin-walled, but those of the plant growing under the adverse condition are narrow and thick-walled. These are living cells, however, chloroplast is not found in them, except in the aquatic and allied plant. The outer radial and tangential wall are commonly impregnated with a fatty and waxy substance called cutin, and the deposit layer is referred to as a cuticle. At place, the epidermis remain interrupted due to the presence of stomata in the grain stem. In certain cases, epidermal cells also give rise to various types of multicellular hair, also called trichomes. Epidermal is protective in nature, which protect the inner tissue from desiccation, excessive heat, and from microbial attack. Due to deposition of cutin, it highly minimizes the loss of water due to transpiration. It may persist throughout the life of the stem, as in the case of ephemeral and herbaceous plant, or may be destroyed or slouched off as a result of secondary growth and the formation of peridium, as in the woody plant. Ground tissue system. In dicotyledonous stem, the ground tissue remain differentiated into hypodermis, cortex, and pith. Such a differentiation is not found among the monocotyledonous stem. Hypodermis is herbaceous. In herbaceous stem is usually collenchymatous type, which may be in the form of a continuous ring, as in case of Aristolochia, or in the form of a patches in annular stem such as cucurbit and peristrophy. Chloroplast may or may not be present in these cells. Their primary function is to provide mechanical strength, and photosynthesis, if found, is a function of secondary importance. The cortex is often parenchymatous and consists of thin-walled, round or oval cell with a large number of intercellular spaces. Outer few layers of cortical cells may be assimilatory in nature due to the presence of chloroplast in them. The chlorenchymatous cortex is well marked in the phylloclade stem of Euphorbia tiruchelli. As in the case due to the absence of leaves, stems take the function of leaves. The cortex in most of the aquatic plant mainly consists of parenchyma with a system of large intercellular spaces. In certain plant, lactiferous cells or duct, mucilaginous canal, resin canal, tenile cell, etc. may also be found. The innermost layer of the cortex is called endodermis, which consists of cell with a Casparian strips thickening. However, in certain cases, the cell of the innermost layer are rich in starch grain, and then this layer is called the starch sheath. Vascular system. The most important constituent of the vascular system is the stele, which consists of vascular bundle. But in addition, pericycle, pith, medullary rays may also be found in this system. Pericycle. The tissues lying inner to the endodermis and outer to the phloem are collectively referred to as pericycle, and these are regarded as a non-phloic in origin. Pericycle is distinct and well marked among dicotyledonous stem. Often it consists of sclerenchymatous fiber, which may be found in the form of a solid spherical bands as in cucurbita, or in the form of patch as in Helianthus and medicago. Vascular bundle. The stele of dicot stem is polyfascicular siphonostele or dissected siphonostele, consisting of vascular bundle arranged in the form of a ring and interstellar ground tissue. The vascular bundle consists of primary phloem, cambium, and primary xylem. Xylem is end arc, protoxylem lies toward the center, and metaxylem toward the periphery. In the young stem, the vascular tissue differentiates from the procambium, also called the provascular meristem, in the stem apex. The primary xylem is formed centrifugally, that is, protoxylem is formed first and the metaxylem later on. The cambium found in between xylem and phloem is the persistent residual meristem, which is called the intrafascicular cambium. The vascular bundle in general are conjoined, collateral and open type. However, in cucurbids, these are conjoined, bicollateral and open type. The open type of vascular bundles are capable of undergoing secondary growth. The stem are usually monostelic type, but the polystelic is found in the rhizome of several members of Nymphaceae, where each stele consists of 2 to 20 or even more vascular bundle. Pith. Pith forms the central part of the stem and also the stele, which remains enclosed by the vascular tissue. It usually consists of loosely arranged parenchymatous cell with abundant intercellular spaces. In some cases, in case of cucurbita, pith cells are destroyed and a hollow pith cavity is formed. The cell may store starch and sometimes waste product. Lactiferous secretory cell, sclerites, idioblasts, crystals of various kinds, and eragastic substance may be found in pith. The outer few layer of the pith may be composed of small, thick wall cell, and such a distinct outer pith is called the perimedullary zone or the medullary sheath. Medullary rays. These are the narrow, vertically elongated bands of thin wall parenchymatous cell, transversing from the pith toward the periphery. In cross section, they look like radiating bands of parenchyma separating the vascular bundle. Their main function is the lateral or transfer conduction of food material and water. Sometimes these periderms are the function as a food storage. Medullary rays are very prominent in Aristolochia. Thank you.